Okay, it has been 30 minutes and we are ready to look at our cells, see how well they've grown on their different materials. Culture hood. Okay, so the first thing I need to do is wash off the cells and stuff that don't want to be there. We're only interested in the ones that have actually formed some sort of stable attachment. And so there will be some that are just kind of on there, and we don't want to count those. So we're going to wash them pretty good with PBS. So the first thing I'm going to do is suck off the cells and the media. Again, we're very not concerned about sterility here. I mean, our experiment is essentially done. You know, I could even realistically just do this on the bench top because it just doesn't matter anymore. Okay, so we only put 800 microliters in each. I set the pipetter to 1 ml just to, you know, make sure that we're going to get it all off. It makes it easier just to have that little bit extra volume in case I suck some air up. Okay, I am going to change pipette tips in between the cases because I don't want to contaminate like any cells from one to another. Okay, now when adding the PBS, we could do this just with the, you know, a single pipetter and just kind of try to get about 1 ml into each one. It doesn't have to be completely perfect. But we're just going to do this wash step twice. Okay, I'm going to kind of shake it around. We just want to agitate these cells enough because we don't want them just sitting there. If they don't want to be there, this is your time to go away. Okay, I'm going to keep shaking as I suck up just to make sure that all the cells have the opportunity to go away. All right, so you'll notice there is some red-ish color, right, in these gels with wells, and that's because some of the liquid does get into the gels and kind of hydrate them, which is totally fine, and then it just retains the color a little bit. So it's not that big a deal. Okay, so now we're going to do the wash step one more time. All right, this time I'm trying to get like almost all the liquid off. So that cells that are not stuck, that are maybe suspended in the liquid, do not have a chance to like look like they're stuck. Right, we only want cells that have attached the gels to be there. Okay, and again, change my tips between cases. Okay, and so I could go ahead and like add just a little PBS to each of these wells to kind of make sure they stay hydrated while we take the pictures. But I'm going to take the pictures pretty quickly, within like 5 minutes or so. I'm not going to let them dry out. So I'm not going to worry a whole lot about that. Okay, so we are done in here. Like I said, I'm going to go quickly. Let's go take some pictures before these things dry out. Okay, so one of the challenges in this particular experiment is finding the top of the gels. And it's actually really hard to do. And you can really only do it if you can find some cells. So I'm going to kind of show you what this looks like. First of all, I've cranked the microscope focus all the way down. So it's like the objective is down as far as it can. So as I raise it up slowly, the first thing we're going to encounter is the bottom of the well plate. And it's usually pretty easy to find because it's nasty and dirty. Alright, and so there we go. We're starting to see some nastiness from the well plate. Just those specks and streaks and everything else you might see. So I'm going to continue working my way up, and I'm going to get to under the hydrogel. And you might see some debris or a cell or two under there. Like maybe that piece of black in the middle is a little piece of debris under the gel that comes into focus. And then we keep coming up, and we finally start to see some cells. Here we go, they're on top of the hydrogel. Okay, and so now this is pretty good confidence that we've reached the top of our hydrogel. And again, this is something that's really hard to do. You just have to get practice on how to use the microscope. So we crank it all the way down, and then we kind of raise the focus up through it until we finally find it. So I'm going to take some pictures of all the wells and let you count them and figure out which one has best attachment. Okay, so we are done taking pictures of these. You guys will analyze them. There's not a ton of cells. I could barely find any, if any, in the agarose only. I found a few in the agarose with gelatin, and then you saw plenty in the controls. You can see the control pictures were kind of funny. So it's one of the, I guess, things that happens when it's just dry, and the bottom could be refracting light a little bit differently, and just like the light could be bouncing off the sides depending on how close to the sides I was. The pictures look a little bit different and weird, and so hopefully you can see the cells. I could definitely do it when I was looking through the binoculars and see cells. It's a little bit harder when I was trying to look at that small camera screen to make sure I captured what I was seeing in the camera as well, and they have different focal lengths, so I couldn't do the exact same thing I was looking at and take a picture. But you guys will be able to see the pictures of what happened here and do the analysis, and that will be it for the cell culture experiment.